I think we finished this section on antibodies. I just wanted to refresh what we're talking about. Plasma cells. Making antibodies requires a first exposure. They have to be sensitized. That takes a little while, 5, 6, 7, 8 days. But once they've been sensitized, they start cranking out antibodies. Technically plasma cells that are doing it, but they're a type of B lymphocyte, so you can call them B lymphocytes, that's fine with me. And then this is our sort of model of what an antibody looks like. The key of an antibody is that there are very specific sections on the antibody that can bind to a specific antigen. So the key here is specificity. One antibody is going to bind to one type of antigen, and theoretically only one type of antigen. Complement system. So before we actually hit the complement system, and I know that's the subheading, this is not part of, well, this is not all part of the complement system. Opsonization. This word opsonization refers to the ability of antibodies to tag specific antigens for destruction by other types of white blood cells. So if we can imagine a bacterial cell, and it's got antigens on its surface, I guess I made my antibody really like way too big. Here's my antibody. The plasma cell has released it. It randomly floats through the circulation, comes out into the tissue, and then can recognize a specific antigen on that cell, and then it binds to it. And what that connection does is it tags this particular pathogen, this bacteria, for destruction. What's going to destroy it? Anything that can phagocytize. What are our two types of phagocytic cells? Macrophages and neutrophils. And essentially what these cells are doing are recognizing, hey, there's an antibody attached to something, let's eat the whole thing. So it can identify it and then snuggle up to the whole thing. Let's give it a U-shaped nucleus, recognize it by its own receptor proteins and say, hey, there's an antigen and it's bound to something. Let's engulf this entire thing, put it in a vacuole, put digestive enzymes on it, and then destroy it. So this is really convenient because if you can make an antibody versus something, you can tag it for destruction. And you've got wandering macrophages that are just basically looking for things to eat. They find anything with an antibody bound to it, then they eat it and then destroy it, which is fantastic. And neutrophils can do the same thing. So that's opsonization. This word is weird. I should really look out where they came out. There's a lot of things that are referred to as opsons. They all have to do with antibodies and things that antibodies do. So the complement system works in addition to this opsonization. So this is really the complement system. And this is weird. Like this part makes sense to me. Tag things for destruction. Perfect. What the complement system is utilizing a whole set of proteins that are already in the blood and can filter out into the tissues to have other effects besides this tagging things for destruction. So here's a nice example from your textbook that shows here's one antibody that's bound to a bacterial membrane, for example, and then right next to it, there's another antibody that's bound. So the close proximity of these two things right next to each other actually allows other proteins, complement proteins, to bind to this complex. So the complement system is going to rely on proteins. And there are lots and lots and lots of different complement proteins. We don't need to list all the different types. All we really want to do is list some of the functions that these complement proteins can have. So one of the things that these complement proteins can do is release chemicals that attract macrophages. I should say phagocytic cells because it could be neutrophils, phago. In other words, they can start to lay down a trail of molecules that wandering macrophages can run into and say, oh, hey, there must be some antibodies bound to something nearby, and then they can hone in on where it's coming from and then phagocytize it. The other thing that they can do is change proteins that are different complement proteins and turn them into things that make pores. And those pores are inserted into the membrane. So we could say something like cause proteins to form pores. I'm going to call them large pores. So if you imagine the plasma membrane of a bacterial cell, and then in it we insert one protein and then another protein, and then another protein, and then another protein in kind of a circle, but that there's a space between them. That allows things to go into the bacterium. That also allows things to come out of the bacterium. We saw something similar to this before, right? We looked at natural killer cells. We had those perforins that perforated the membrane. It's similar. They're not the same proteins, but it's a similar concept. If you start punching holes in a cell's membrane, it's going to spill out all its contents. Things like sodium and other molecules are going to go in, and hopefully that's going to kill the cell. There was one more thing that they do, which I forgot. Inflammation. So some of the complement proteins that are activated by the system cause inflammation. What are the types of cells that cause inflammation in your tissues? Mast cells? In other words, they activate mast cells. And what were mast cells releasing that causes inflammation? Histamine. Right. I heard it. Excellent. Your book has some other functions. I think that's pretty good. It's a nice list of functions for this complement system.
Does that include all kinds of inflammation? Like there's no specific kind of inflammation?
Inflammation is a pretty general response.
Okay.
Yeah. So yeah, it's right, a general response. Venules increase the size of the pores, fluids come out, irritates nerve endings, make things itchy, make it hurt. Yeah. Anything else I can add to that? Have you guys tackled this section on T lymphocytes yet? I see Sonia smiling. Either you tried to tackle it or you meant to and you didn't.
Yeah, I tried to.
You tried to read it?
Yeah.
Yeah, what happened when you tried to read it?
It's too confusing.
Yeah.
Yeah.
All right, that was my experience. So I'm going to simplify this section on how T lymphocytes operate. It really is complicated. I think it's a testament to what we've been able to do with the amount of money that we've dumped into studying the immune system since the mid-'80s. That we can even figure out what's going on. So I cannot possibly talk about it all. I'm just going to simplify it to some basic concepts that are true. But in the book, lots of detail. I'm just not. So, importance here, we're talking about specific immunity or adaptive immunity, and the B cells produce antibodies, that's specific, and then our T cells, our T lymphocytes, are the other form of specific immunity. So we're talking about the function of T lymphocytes. Let's get this. And B lymphocytes used humoral immunity. They were specific, but they secreted antibodies into the blood. T lymphocytes are going to directly attack cells, so this is cell-mediated. So the main type of T lymphocyte is the killer T, sometimes called cytotoxic, sometimes called CD8, sometimes with a plus cell on it. Of all these, I like killer T the best. Killer T is an excellent name, and I always, maybe I'm showing my age here, but I remember the Terminator movie came out. The other one, the first one was the best one. I don't care what anyone says.
The second one.
The second was fine, but the kid got annoying. Killer T cell, the Terminator, one specific mission, right? He comes back in time, he is looking for one person that he wants to kill, right? That's it. He might kill other people by accident. We're going to ignore that. He was after Sarah Connor. Killer T cells are specific. Once they learn to attack one specific type of antigen, that's really all they're going to do. And they literally go out and they try and find it, and they got to get their hands on it and then kill it up close and personal. So that's what our killer T cells are doing. Up close and personal, cell-mediated immunity. They're going to directly attack cells. Directly attack. And I'm going to say antigen-bearing cells.
Listen.
In other words, the reason that they're able to identify a particular cell is that on its surface, we have to have the antigen that they've learned to recognize. And what they can do is recognize, hey, this antigen's on this cell, we're going to destroy this cell. What types of cells are they going to directly attack? Mutated cells. If you have a cell that is mutated, it might produce mutant proteins. In other words, DNA codes for proteins, DNA gets mutated, it might be making a protein that's really weird. That protein can be recognized as an antigen, so that's one sort of cell.
Like, how good are they at attacking it? Like, is it just, like, how good are they at attacking? Do they just like, immediately get rid of it? Just like,
We'll go over that.
Oh, okay.
And it'll ring bells. Virus-infected cells. Viruses that have infected cells are producing their own types of proteins. So they've got their own DNA or RNA that is used to make proteins inside the cells. Those are foreign proteins. Those can be recognized as antigens. So here's sort of the two big categories of types of cells that T lymphocytes are going after. Those that are mutated, those that have viruses within them. Mechanisms. So mechanism of destruction. Here's our cell with the antigen on its surface. Our T lymphocyte recognizes it. Hey, that's a foreign antigen. That's foreign. What's it going to do? Two main processes that it can use to kill these cells. First, inserting perforins. Perforins are proteins that once they're inserted into the membrane, self-assemble into little tiny pores. In other words, it perforates the membrane. And if you've got little tiny pores, things can go into the cell, things can go out of the cell. Second thing that it does is it activates enzymes initiate DNA destruction and apoptosis. They insert granzymes, enzymes that once inserted into the cell activate enzymes that go into the nucleus and then destroy the DNA. They also activate enzymes that initiate apoptosis. Did we talk about apoptosis before? No. Apoptosis is cell self-destruction. So it's the ability of a cell, and all of our cells have this ability, to basically take their lysosomes, which contain digestive enzymes and acid, and then to release the digestive enzymes and acid into the cytoplasm. If you start dumping out all the acid and digestive enzymes into the cell, you're going to destroy everything that's in the cell, so all the organelles as well as the DNA. We've seen this before. Inserting perforins, inserting granzyme. Natural killer cells do this. So they're not really doing anything different, it's just a different way to recognize the cell. Whereas a natural killer cell is looking for cells that lack the right self-tag. Here our T lymphocyte has been trained, if you want. It has learned to recognize a specific antigen. If it has that antigen, inserts the perforins, inserts the granzyme, and then hopefully that destroys the cell. If it's a virus-infected cell, hopefully it destroys it before it reproduces lots of little virus particles and releases those. The role of helper T cells. I'm going to just make a statement and then not talk about it. It's not a bad name. Helper T cells. Sounds like they might help. They secrete numerous chemicals that help activate T lymphocytes and also help activate B lymphocytes. So you can read about what they specifically do, but I'm simply not concerned about it. It's too much detail. They help. They're an integral component of activating T lymphocytes and B lymphocytes. So this is how T lymphocytes work, killer T cells. We haven't covered how it is that T lymphocytes become activated. How do they learn how to attack a specific type of antigen? So if we look at a T cell, it has a protein on its surface, lots of proteins on its surface, that are essentially a place that they can bind antigens. It's a place where foreign molecules that are found can be placed. But in order to get those antigens here, they have to be picked up by another type of cell. They rely on dendritic cells. And you may or may not have seen dendritic cells when you studied the skin in your anatomy class. They're basically a type of wandering macrophage. And they also can get them from macrophages. Moving around in tissues, looking for basically dead cells, looking for cells that are not quite right, dead, dying, virus-infected cells. Our cells these are viral particles inside a cell. The cell has lots of viral particles in it. It's dying. We're going to have it sort of bursting open so that it's sort of fragmented and dying. Macrophages can recognize lots of types of material, including cells that are dead and dying. The macrophage eats this entire thing and is able to pull out these strange particles, viral particles, and it puts it on its own class of protein. This is a type of MHC, major histocompatibility protein, and once that viral particle is on the surface of this cell, it puts it on the membrane surface itself. So in other words, what this has done is eaten a virus-infected cell that's full of weird types of proteins, bound those proteins, put them on its own special MHC protein, and put it on the surface of its cell. Now it basically has a copy of the potential antigen. It goes to a lymph node, it goes to the spleen, it goes to some malt, and it finds a T lymphocyte and then presents it. And essentially, we can join that particle to that MHC protein on our T lymphocyte. Now a T lymphocyte has the ability to recognize any cell that has that protein on it. So it's not a, let's say, the cell itself hasn't really learned to do anything. It's simply getting a copy of an antigen that some other cell has found and said, this thing is foreign, gives it to it, and now the T lymphocyte says, all right, this thing is foreign. I'm going to go out and find something that looks just like this, and when I do, I'm going to kill it. So out into the tissue it goes, looking for cells that bear that same antigen. And when it finds that same antigen, it assumes that cell, it must be a mutant cell, it must be a virus-infected cell, and then it destroys it. So you might ask the question then, if this is a quote-unquote normal body cell, how does the antigen get on its surface? If it's a yeah, I mean, we have, if it's a virus-infected cell, the viruses are going to be inside. Our cells have an innate ability, a natural ability to produce their own proteins, MHC, major histocompatibility proteins, histocompatibility, that take up chunks of proteins, all kinds of proteins, busting up our cell proteins and then putting them on the MHC compounds and the complexes and putting them on the surface. Most of the time, those are our own proteins. It doesn't matter. But if it's a virus-infected cell and it's just sort of grabbing chunks of random proteins, putting on this thing and putting them on the surface, it might grab a virus protein. And then that gets on the MHC, and then that gets on the surface. So the cell's just going about its own business, putting on chunks of stuff, putting it out there. As long as it's our own proteins, it's not a problem. But if you're infected with a virus or your cell is mutated and you're making weird proteins, something strange has gone on the surface. That gives your T lymphocytes the chance to recognize it. And then they can go through inserting their perforins, activating the enzymes that cause DNA destruction, apoptosis, inserting those granzymes, and then destroy the cell. So the keys for T lymphocytes are, it's specific. They have to be presented with the antigen, but once they've seen it, then they can go out and kill many, many copies of cells that have it. Every time they bump into one that has that same antigen, punch holes in it, cause apoptosis, and then destroy the cell. Doesn't work against all types of pathogens. Works really well versus virus-infected cells, and if you've got a mutant cell. Could be a tumor, for example. Good. Your textbook goes on for about 4 pages, a lot more detail. I'm trying to keep it at this level. So you can tackle it if you want. Take some of his advice. What did you say? It was complex. For that section of the chapter? What did you say? It was complicated?
Confusing.
Confusing. That's a better adjective. Yeah. So keep it the basic principles, what the T lymphocytes are doing, and it's always specific. All right, let's talk about active and passive immunity to finish off this chapter. So let's define them first. Active immunity, a person's own defense mechanism. Lymphocytes. T lymphocytes. So that works. A person's own B lymphocytes and T lymphocytes, their own adaptive immune system, is recognizing and destroying the pathogens. It's the person's own natural cells that they possess in their bodies that are doing all the action. Passive immunity is relying on antibodies that come from another source. Source, I would say person or animal. In other words, in passive immunity, we require some other player. It could be someone with a needle. It could also be someone like your mom when you're an infant giving you antibodies through breast milk. But the immunity that is imparted on the individual is not your own immunity. It's antibodies that come from some other choice. So really we only see this in our lives in two places. One, from another person, coming from mom in breast milk or colostrum. What type of antibodies are going to be in breast milk or colostrum? IgG or IgA? And an animal. What do you mean by an animal? Like, are you giving animal antibodies and putting them inside your body?
No, because I seen one where like a human and a bird virus combine, and the pig gets it, and then the pig could give it back to the human. I saw that in my bird.
It's true, but we're not talking about that. Different type of animal-human interaction. Let's say you're. Let's try to give a good example. Perfect example. You are bitten by a rabid bat. We'll do that one. It's a different one. So we'll do it. And venom's also good. But you're bitten by an animal with rabies, or it might have rabies. So now you've got the rabies. It's a virus. It's a rabies virus. It's inside your blood. Do you have time to gain your own immunity? That is, go through. No, you don't. You have to tackle that virus and whatever virus-infected cells immediately. How are you going to do it? Well, you're going to need antibodies. Where are you going to get them from? We get them from animals. So there are animals somewhere in the United States that are injected with attenuated rabies, and they have their own immune system which can recognize the rabies virus very easily. So if you inject some rabies into one of those animals, they respond by producing many, many, many antibodies. We take their blood serum, we purify it, we get their antibodies. It could be from a goat or a rabbit or like a horse or something like that, and then we take those antibodies that the plasma cells of that goat made and we inject them into you. Once they're inside you, they're just antibodies. So even though maybe it came from a goat, it can still float through your plasma, identify the virus or the virus-infected cell, attach to it, mark it for destruction, or activate your complement system. And hopefully what that does is it gets rid of all the rabies viruses that are now in your blood and that you don't get rabies. Passive immunity, because these are antibodies that are transferred into you from another source. If you get bitten, by this rabid bat and you go in, and they used to be a series of like 7 injections. It's not that bad now. I don't actually know if they have to go into your stomach anymore. It's a little bit better. You go through your treatment. You're rabies-free. Do you have any immunity to rabies? No. And you usually don't. If you get the treatment of antibodies, you never allow your immune system the chance to recognize, oh hey, these are rabies viruses, and I will have my own response to it and develop plasma cells and memory B cells that can produce our own antibodies. So having this passive immunity, a transfer of antibodies, typically doesn't impart on the patient or the person any of their own natural ability. What if you wanted to be immune? What if you wanted to be able to fight off rabies in case you got bitten by a rabid bat? Then you would need to get a vaccination or an immunization, and then you would get probably a series of shots. I can't remember if it's 1, 3, or more. Attenuated or dead rabies virus particles injected into you. You don't get rabies because you're being injected with something that's dead, and then you allow your B lymphocytes to recognize it, produce plasma cells, and produce memory cells so that now you have an immunity to it. Then if later, 2 months later, 3 months later, you get bitten by a rabid bat, you have your own active immune response, produce your own antibodies to destroy it. Kathleen, you mentioned antivenom. So like if you're bitten by a rattlesnake, for example, this is the kind of case. It's like, well, it would be nice if we could be immune to rattlesnake venom, and theoretically you could build up some kind of immunity to it, but if you've just been bitten by a rattlesnake, you don't have really time to mess around. So there are antibodies that are produced from animals that use rattlesnake venom and inject them into horses, collect serum, purify the plasma, get the antibodies, and then inject that into you. Same with spiders and scorpions, yeah.
I saw a guy got bit by a rattlesnake, and then there was a deer close by, and they shot the deer, and they took out the heart, and then the beating heart, like, he put it on his skin where he got bit, and it took out the venom. Is that true?
That's nonsense.
That can't happen?
And in fact, when you read about first aid for rattlesnake bites, they tell you not to cut and slice on it or suck it.
No, it was like the deer's heart. They took the deer's heart and put it on the arm where he got bit.
No, it doesn't make any difference. Hopefully they got some good steaks out of it, but
I don't think the deer had a baby, so that's the deer baby.
Is the name Bambi?
We went over this already.
With everything we see on TV.
if it appeared to work, it worked because the rattlesnake didn't actually inject any venom when it bit the person. So yeah, I'm sure it appeared to work great, but
Too late, Bambi's
That's
there.
great TV though. That's great TV.
Killed the mom, and then there's a baby.
rabies, snake venom, that's a good one, and there's some other ones too. Somebody, you step on a syringe needle, like that's on the street, and it pokes through your skin. You don't have time to go in and get like hepatitis vaccinations. No, you go in and you get antibodies to get your passive immunity so that you don't come down with hepatitis, for example.
You mean if you can walk there?
Well, sure, it's the beach, maybe. Somewhere you kick your shoes off. Ow, I just stepped on a syringe.
Never going to the beach
Could
again.
happen. Yeah. Or you never know, maybe someone spits blood in your eye, you know? You don't know what's going on. I mean, you're going to be a nurse. You'll be surprised what flies. All right. Yeah.
Don't go to rapes. Don't do it.
All right. Questions on immune system? Yeah.
Yeah, my
Probably.
brain's a little fried.
Probably. The deer heart, really? Deer heart sucking out the rattlesnake venom? Nah. Nah. Not buying it. Yeah, Tisha. Underwear? IgA. Immunoglobulins. The type of yes.
I'm so tired right now.
Good for,
Isn't it like
Yeah.
Emergencies.
Yeah.
Yeah, it's great for emergencies. In fact, this was the very first type of treatments that we even had. Before we even really knew what any of these things were, they found out that if you had a particular disease, you could take some of your blood, inject it into an animal, allow them to develop an immune response to it, then inject some more, and they'll have a big immune response, and you can harvest their antibodies. As long as the person could stay alive through that 2-3 week process, then you could do it. Yep. And they used to use horses, because you could get a lot of blood out of a horse.
caffeine doesn't do anything to me? I like taste coffee. Which is kind of good, but kind of bad at the same time. Sometimes I like a little energy boost.
You guys got 2 shots. 3 shots. Really?
I can drink coffee at like, you know, o'clock at night. Totally fine. Oh god, repro.
A lot of things just to start with interest. All right, the reproductive chapter, it starts off slow. A whole section on basic reproduction. Sperm, eggs, meeting of sperm and eggs, number of chromosomes. We've covered a lot of that. Just read over it. I really want to jump into endocrine regulation. First basic principle of our reproductive system is it's basically entirely driven by hormones. It's an endocrine-related function, and it starts at the hypothalamus. Hypothalamus is responsible for regulating the activity of the anterior pituitary, which it does via hormones, and the anterior pituitary regulates the gonads. If you're a female, there's one more step in here, which is the gonads regulate what's happening in the uterus. So this is really kind of a top-down. Hypothalamus affecting the anterior pituitary affects the gonads, gonads affects the uterus. And it's also negative feedback, so we have negative feedback loops among all of these structures. Basic functions, and we should know these, and I know you guys know these, but we're going to do it anyway. Functions of gonads. And you guys are happy with this word gonad? Sure. All right. You know that it can mean ovaries and testes. It's not just nads for dudes. All right, good. Main function we know for the ovaries, egg production. What's another function of the ovaries? Besides egg production, production of hormone production. And we have our two main hormones, estrogen and progesterone. There are lots of different types of estrogen. There's one main type, estradiol, that comes out of the ovaries. I don't use that term, I just say estrogen, but just recognize that there's lots of different types of estrogen. Estrogen has all the feminizing effects that we think about when we think of the female reproductive system. So it's responsible for the development of sex organs. It's also responsible for the development and maintenance of secondary sexual characteristics. Sex.
By second, you mean like?
So let's do each in order. For development of sex organs, the vulva, external genitalia, the mucosal lining in the vagina, the breast tissue. All those are sex organs. Secondary sexual characteristics are related to being female, but they're not sex organs. Deposition of fat patterns would be a good example. So women complaining about fat being deposited on their butt, for example, whereas men, well, oftentimes it's not being any deposited in how much food they eat. It all goes like right here, and then they have this tiny little butt. Some of us that's just what we have to live with. So that's one thing. The skin remaining soft versus becoming tough would be a secondary sexual characteristic, for example. So they're not directly related to the sex organs, but they're related to being female, for example. In addition to these functions, and this is really the way to think about what estrogen is doing, we have progesterone that works with estrogen to do two more things, which is maintenance and maintenance of the uterus. And of the uterus for female cycles, but also of the uterus during pregnancy. And there's an interesting complication here is when the developing embryo takes over some of this maintenance of the uterus, but generally it's progesterone heavily and estrogen that are responsible for developing and maintaining the uterus. For males, we can make sort of similar patterns, except for it's a little easier to deal with. We know that the function of the testes are for sperm production, and the testes are also responsible for testosterone. We've only got one hormone to worry about, so we don't have to worry about estrogen and progesterone. We just have to worry about testosterone. But the effects are going to be similar as to estrogen, only now for males. So primary sex organs. And again, this is development and maintenance. In other words, if the levels of testosterone fall, the sex organs will atrophy. And the same would be true for females in estrogen. If the levels of estrogen fall, then the sex organs can atrophy, become less well developed, get smaller, and lose their functions, not just structural. And also, testosterone is responsible for the secondary sex characteristics. So things like hair growth, muscle growth, bone growth, increased hematocrit, all those would be secondary sexual characteristics. Development. Good. Control of the ovaries. These hormones, estrogen, progesterone and testosterone have powerful effects on lots of body systems. So what the hypothalamus is doing is really it's controlling them, controlling the levels so it's not too high, so that it's not too low. I don't have behavior in here. Really should put behavior. We hear about roid rage. Roid rage is real. If you inject testosterone into people, testosterone does cause aggression. And if you are very aggressive and also irritated, you can fly off the handle. And it's purely testosterone. So behavior actually should be included as a secondary sexual characteristic.
Does it become solely from testosterone?
Not so.
As opposed to
Well,
like
you-
actual steroids? Like you're
Yeah, so when people take steroids, that's the big one. There are other steroids you can take, but testosterone is the granddaddy
But if you-
of the anabolic
So
steroids.
that's why like people are really angry that they have a lot of testosterone.
You know what? If you know a person who's really pissed off all the time, it would be fun to take a blood sample, see what their testosterone levels are like. It
Is
might
that
be
just for
elevated.
males or just testosterone?
Levels of testosterone in female are incredibly low. There's very low levels, but there are some, and it comes from the cortex, but very, very low. Not guaranteed by any means, but yeah.
Women have different hormones. Makes them crazy.
So we're talking about the control of the gonads. It's the hypothalamus and the anterior pituitary working together that control secretion from the ovaries and from the testes. And then for females, we also have to talk about the role of placental hormones. Fetal hormones. Because placental and fetal hormones can influence secretion of hormones from the ovaries. But usually it's the hypothalamus and anterior pituitary. So how does this work? I drew this in lab, we'll draw it again. We know that the hypothalamus is responsible for regulating hormone secretion from the anterior pituitary. And when we studied the hypothalamus before, I said, there was some releasing hormones that the hypothalamus secretes and that affects the activity of the anterior pituitary. Now we're going to name just one of these releasing hormones, GnRH, which stands for gonadotropin releasing hormone. So gonadotropin releasing hormone is a hormone that is released into that portal system, travels to the anterior pituitary, and it stimulates the secretion of both of our gonadotropins, our hormones that cause the gonads to become active. That is, it causes the secretion of LH, luteinizing hormone, and FSH, follicle stimulating hormone. Both those hormones now in the blood, they travel to the gonads, and cause two things to happen. One, the gonads are going to secrete the sex steroids, which is going to be estrogen and progesterone for females, and for males, testosterone. It also causes the production of gametes. How does the hypothalamus know, and let's do males just for simplicity's sake, that the levels of testosterone are appropriate? That is, hypothalamus secretes some GnRH that goes to the anterior pituitary is cranking out LH, levels of testosterone are going to start to go up.
Do you mean like chicken?
Yeah, so the levels of testosterone are going up, going up, going up. The hypothalamus possesses sensors, and the sensors detect the levels of testosterone in the blood, and then they negatively impact it. In other words, testosterone has a negative feedback inhibition effect on the hypothalamus. It causes the hypothalamus to decrease the amount of GnRH that's being secreted. It also has a negative feedback effect on the anterior pituitary itself, so it directly inhibits the anterior pituitary. If GnRH levels are going down, LH levels are going to go down, and FSH levels are going to go down. The gonads are going to be producing less testosterone. The levels of testosterone will fall, and as those levels of testosterone fall, our negative feedback inhibition is going to be removed. And now that the hypothalamus and anterior pituitary are not being inhibited, they crank up their production again. Out comes more GnRH, more LH, more FSH, and then the gonads are producing more testosterone until the levels get high enough again, and then it inhibits them, once they're inhibited, less GnRH, less LH, less FSH, and the levels of sex hormones are going to decrease. So this is classic negative feedback inhibition. It's a simple process, basically where the product that we're ultimately making, the sex steroid, estrogen and progesterone, and testosterone, inhibit the very originators of the system, GnRH secreting from hypothalamus and LH and FSH from the anterior pituitary. Is that part okay? You all right with that? Yes. So the hypothalamus in this case would act as a sensor, and the anterior pituitary also acts as a sensor, as a sensor, since it can be directly inhibited. I'm not going to talk too much about pulsatile, but the way that these systems work is not with a constant level of GnRH secretion, but there'll be a pulse of GnRH, like a blast, which releases GnRH, and then we have a response, but then there'll be a pause, and then another blast, and then a pause, and then another blast, and then a pause. And you think, well, that's really weird. Why would we have a sort of a fluctuating, random, or pulsatile release of GnRH? Probably the best way to describe it is what happens if you do something to get rid of it. And the best way to get rid of a pulsatile release of hormones is to just inject them inside you. So people who inject testosterone for growing muscles have a very high level of testosterone, which results in not a pulsatile negative feedback influence, but a continuous negative feedback influence. In other words, if we're not getting our testosterone from the gonads, that is, it's coming from a syringe.
Doesn't the body like actually stop making testosterone if it's always coming from the injection?
Our negative feedback is not going to be pulsatile either. Our negative feedback is going to be constant. And this downregulates the number of receptors that are present on here, and it really, over a long period of time, downregulates the whole mechanism so much that the negative feedback actually destroys these organs, and they no longer produce any GnRH. Even if you get rid of the syringe, you decide, I'm big enough, or I'm 40 and I'm not going to lift weights anymore. And without any GnRH, you're not producing any LH, you're also not producing very much FSH. What's going to happen to the normal amounts of testosterone that you're producing? You can't do the zero. So, but nobody else. Yeah, so if you did that with estrogen, I don't think the effects are as prominent, but you can imagine a situation where maybe a person just takes a lot of the glass pills or something like that, you might have the same thing. But the estrogen pills are cyclic. You know, have levels that increase and then fall, increase and then fall. No, it's not enough in there. People who've done this have a patch that they will have to wear, because remember what testosterone does: maintenance of sex organs, size and function. So not only are they, big in muscle, and then they stop taking testosterone, and then they start getting smaller, other things start getting smaller and basically stop functioning altogether. I'll get your question in a second, Tony. This is a weird story. This is like back in the day, and I'm like the last generation of students who went through graduate school who actually had to go to the library and like get magazines and like articles and stuff. And when you end up there, then you end up just like looking at random stuff. And there was a study that was done with the effects of testosterone. What they did was not ethical. They took people who were obviously mentally retarded, who had undeveloped testes, so that they were not producing their normal testosterone. And then they injected testosterone to see what would happen. So they start off with the pictures, the faces are all blurry, you can't see anything, but their external genitalia are there, and they start off, genitalia are basically the size of children. They're not developed. We don't have testosterone, we don't have the development portion, and then over the series of months of these injections, the size looks pretty normal, and then they stop injecting it, and then they got the series of plates where the size is going all the way back. So you got the start, all the way up, and then all the way down, and it's pretty much back where it started from just an endogenous injection of testosterone, which I know for a fact that if you look at the years and the things that they did to people back then, probably not.
Say injection will cause the anterior pituitary and will the GnRH not?
Yeah, over a long period of time. So if, if this happens in the short term, your own endogenous testosterone production goes down, but when you stop taking the injections, they can recover, and then it will go back to normal after a variable amount of time. This is people who've been injecting for 20 or 30 years, and when they stop, the damage is done, and apparently it never goes back to normal.
So if you're gonna inject, don't do it for 20, 30 years.
Show me a better idea. Just don't. Just don't. I don't know. Yeah.
How does that affect estrogen levels, guys? Like the testosterone?
So there should be very, very little estrogen. I avoid talking about estrogen in males. There is a very low level of estrogen, and most of the estrogen actually, it's necessary, it's there, but since there's so much more testosterone than estrogen, the estrogen has very minimal effects. It's counteracted by the testosterone. If the testosterone goes, and now all you have left is the estrogen, then you can have feminizing effects. So development of breast tissue, for example, would be one type of effect. What's that?
Like the testosterone cancels the estrogen.
Yeah. Yep. We don't have time for this. 60 seconds I can tell the story. Old man, like 70 years old, grew a breast. And it's not like a man boob, like a breast. It's a female breast. Got the nipple, got everything on it. Goes to the doctor, and they're worried that you've got a tumor somewhere producing estrogens. It could be, in the cortex or something like that. So they excise it, cut it off, get rid of it. Like a year later, grows other breast. The doctor's like, there's nothing wrong with all your organs. Everything's fine. I'm going to find out. So he goes to the house, and he gets every single medicine that was in the house, and he came up with a yeast infection treatment that his wife was using, and they put estrogens in the yeast infection treatment. The low amounts of estrogens that were in that yeast infection treatment was enough to cross through the penile tissue and get into the blood, and because he had basically no testosterone, that estrogen was enough to have feminizing effects and cause him to grow breast tissue. Because he had no testosterone of his own. So it turned out that she was using it as a lubricant just for whatever, I don't know why, but she just obviously had a interesting life.
Ran out of lube.
It worked. She must have liked it though. Maybe it was. But they put the estrogen there, and it has effects on her tract, so it actually would help maintain her mucosa and stuff. So maybe
She's
she
like
liked
two
it, but
for
she didn't
one
know
deal.
why she liked it, or I don't know.
2 for 1 deal?
Maybe.
Got 2 birds with 1 stone.
Guess so. See you Friday.
See ya.
Shit, I don't want to keep whichever. Let me see.
Solved.
Sorry.
You like that
It's called aerobic, and then some with certain pH levels, like will come out the